Uh, what is central dogma? Central dogma is a term that is used to explain the process of how the genetic code, which is within the DNA, is expressed to manufacture a certain protein. All organisms, whether they are plants or animals, are very heavily dependent on proteins. As you may recall from the previous section earlier in the semester, we learned about the different roles of proteins. Proteins can be structural, they can be enzymatic, they can be used as receptors, they can be used as cell recognition. So there are lots of different need within the cell of a protein, and we as humans are pretty much expressions of proteins. All proteins are also called as, we call them as polypeptide. Polypeptide means poly is many and peptide is the bond between the amino acids that comprises a protein. So they are called as polypeptide chain. If you recall, each of these protein chains were made up of the building block, which were called as amino acids or amino acid. Now each chain corresponds to the nucleotide base sequence of a gene. What that means is that there are altogether 20 amino acids that are present in nature. And we learned in the previous section that there were only 4 nucleotide bases. So somehow the 4 bases code these different combinations of amino acids. And in this chapter we will learn how this whole process is carried on. The nucleotide sequence is what we call as the gene. So in earlier section, which we talked about the different nucleotide sequences such as adenine, guanine, cytosine, thymines, they somehow have a code in them, and we will explore that shortly. Now this image that I've shown over here pretty much highlights the entire process of central dogma. Central dogma begins when you have the first set of code copied from a DNA to what is called as the messenger RNA. Now these messenger RNAs then leave the nucleus, comes into the region which is the cytoplasm, and attaches to a region over here which is called as the ribosome. And when it reaches the ribosome, it sort of starts entering from this direction and leaves from the other direction. So on one side the chain is just floating around the ribosome, but it then gets transcribed, translated into another long chain, which is the protein. So this is in short the entire process of central dogma. And now let's look at each step at a time. The first step of central dogma is a process called as transcription. Transcription is pretty much an enzyme that makes a copy from the DNA. So in old times, for example, if you had a very special book that had to be copied and sent out to all the small cities and the villages, then the person who would transcribe the information would be an enzyme, and the process would be transcription, making a copy of a specific region of the DNA. And the copy that is made is a single strand, and this single strand is called as an RNA strand, and in particular it's called as mRNA. M stands for messenger because it's carrying the message from the nucleus to the outside. The specific role of an enzyme polymerase is to help assemble that mRNA. Now the RNA nucleotide, the thing to note over here is they are composed of adenine, guanine, cytosine, and in place of thymine it has uracil. So everywhere, if the sequence was, for example, TTAC, and this was the DNA sequence, the mRNA sequence that would be formed by this enzyme would then be in place of T, it would take in the opposite of T is A, the opposite of T is another A, the opposite of A is T, but it will be replaced by a U, and C would be complementary to a G. This would be an mRNA sequence that leaves the nucleus and comes to the ribosome. In the second step, once the process of mRNA is made, and which has the code, it then has to be translated into a more sensible sequence of an amino acids. Now in this case, you can see how the DNA is initially present there, mRNA is being formed. The first step over here was transcription. Now in translation, each of these codes of mRNA will code for a certain amino acid, and the amino acid, if you notice, are present over here. There's one called as threonine on this block, there's one proline, glutamine, glutamate, and lysine. Each of them are carrying a 3-letter sequence, and this is going to match the sequence on the mRNA. So this is how the correct amino acid comes in, because the amino acid is carried by this little piece of equipment which is called as a tRNA. A tRNA has on one end, it has these codes, and on the other hand it's got an amino acid, and it sort of helps it to align in the right sequence. The sequence again is present in the mRNA code that is present over here. Okay. Now, there's a little bit of difference between a prokaryote and a eukaryote. Prokaryotes do not have a nucleus, and there is no particular separation. So transcription and translation are both taking place within the cytoplasm. In eukaryotes, which are most of the multicellular organisms, there is a distinct differentiation. There's a step over here which is the transcription, the first step, and the second step is carried out in the cytoplasm. I mentioned a couple of times an mRNA, sometimes I call it tRNA. There are different kinds of messenger RNAs. The first kind, which is the mRNA, was helping to code the DNA to the ribosome. The second kind that I used was a tRNA that helped align the amino acids to the ribosomes. The third kind, which is called as the rRNA or the ribosomal RNA, pretty much assembles the ribosomes. But ribosomes are the structures that help in the formation of the protein. So they're outside in the cytoplasm, if you recall. So let's compare DNA and RNA. We did DNA a little in the previous section. DNA was a double-stranded molecule. RNA is always a single-strand molecule. DNA has the information code. RNA pretty much passes this code and also helps sometimes an enzymatic property, which means that RNA can actually also act as an enzyme. And because it has both, it's got the code and it has an enzymatic property, in an evolutionary process, RNA is supposed to be the more precursor molecule, because it had properties of directing a synthesis of a protein and also coding it. Over evolution, it somehow managed to give the role of hereditary information to DNA, and the protein synthesis was given to the ribosomes and direction to the RNAs and so forth. DNA is a double helix, and we already said that. DNA contains deoxyribose sugar, which is a sugar that has one less oxygen. Ribose has a ribose. It contains ribose instead of deoxy. And the last thing is RNA contains uracil in place of thymine. A comparison between DNA and RNA is usually a good question to come up on a test. This is an alternative pathway which I just want to briefly touch over it. Sometimes when mRNA leaves the nucleus, it has a lot of junk carried around with it. What that means is that in all the DNA that we have, there's only 2% of the DNA that makes sense. And humans usually have 3 billion DNA nucleotide, the nucleotides in the DNA. And a lot of this in the region that is senseless needs to be chopped out before an mRNA can be directing a synthesis of protein. And this is called as maturing of the mRNA. And also, this also results in variation among different kinds of sequences. Pretty much DNA can be chopped at different levels, and the introns can be removed. Introns are the regions which makes no sense. So once these introns are removed, the mRNA will be shorter, and it's called as the mature RNA. Now the mRNA also in the eukaryotes is protected by adding a cap or a tail. Just like the cap or the tail is similar to a shoelace. If you can imagine a shoelace without its cap, it will get frayed. So that's how fragile the mRNA is, and it gets all these modifications done before it reaches the ribosome. So what is the genetic code? Genetic code is always a 3-letter word. It's a 3-letter nucleotide that is present in the DNA, and the 3 letters help direct a particular amino acid, and that code is present in the DNA. Now the genetic code that is present in the DNA is copied to a 3-letter sequence, which is now called as the codon. So the DNA is going to have the code, the mRNA is going to have the codon, and then the amino acids will be aligned in a linear manner to form the right polypeptide. Here's a list of codes that are usually present in any biology book that you will find, and each code is a 3-letter code that you can see over here, and each 3-letter code is showing a particular amino acid, a particular base. So each code, this helps an amino acid. The names of the amino acids are written. I'm going to use red over here. These are the different amino acids that are coding by each genetic code. So it's very interesting to see that we have, because we have 4 nucleotides, and if each nucleotide was to code for 1 amino acid, then because there are 20 amino acids, the code would not be enough. If the code was a double sequence of a nucleotide, that is a pair, the code was in pairs, even then we would have almost 16 combinations. That would again not be enough. So the code is now thought of and studied that it is in triplet. There's a code of 3 nucleotides, and each code of 3 nucleotide is going to give you a particular amino acid. But when you have 4 nucleotides, which are A, G, C, and T, and you make 3 different combinations of their possible codes are 64. So that is more than the number of amino acid, and that results in variation among different organisms. So more than 1 code you can see is coding for the same amino acid. For example, VAL is a code for valine, and you have 4 different codes that will go and pick up the amino acid valine. So there's a nice animation that I've also put down for you to watch. This concludes our little mini lecture on the central dogma.